Today is Dr. Christos Papadelis. So Christos is an instructor in neurology at Harvard Medical School, and he is a research associate at Children's Hospital. He's also the manager of BrainMap Laboratory. He did his undergrad, he majored in EE during his undergrad, and he did his master and PhD in medical informatics. He received all his degree from the same university, the Aristotle University of Thessalonica in Greece. And recently he's focusing on his research is focusing on understanding how large-scale cortical networks are formed during human development. So welcome.
Okay. First of all, thank you very much for coming here. It's an honor to give a talk here in front of Dr. Cohen and Matti. I will try to give an overview of my work the last couple of years on MEG, and by trying to put a title on all these kind of things, like I end up with something quite long. I will try to emphasize on two aspects. First of all, the accurate localization that most of the people believe that MEG does not does so accurately, and the functional connectivity. So I will provide evidence that MEG can accurately localize sources in the human brain, and it's also the ideal tool to assess the functional connectivity in the human brain. So I'm pretty sure that most of the people you know these kind of things, but I will give an overview for people who maybe they don't. So the MEG measures the magnetic activity of the human brain. A focal area of neurons, mostly on the cortex, should be almost simultaneously active, and this electrical current generates a magnetic field that is measurable outside from the scalp. The subject usually sits comfortable on an armchair, placing his or her head in a special designed helmet, and this is how we perform the recordings. So compared to other neuroimaging techniques, MEG has an equally excellent temporal resolution, as also has the EEG. In terms of localization accuracy, it lays between the EEG that has worse localization accuracy and the function MRI that's supposed to be the gold standard of the localization accuracy of activity in the human brain. And the most important, people who have used EEG, they regard it as an easy technique. Like you don't have to put the subject inside of a weird tunnel here, or you don't place any electrodes on the head. It's a technique that is quite easy. So an effective and easy way to assess the localization accuracy of MEG is by using phantom constructions like this one, that they resemble the activity of the human brain, and also they resemble somehow the shape of the human brain, or at least the skull. So in this experiment, I used this kind of phantom construction. It was filled with saline solution, and I placed dipolar sources inside that they were activated from electrical currents. The dipolar sources, they were constructed from two gold balls. They were connected with a cable that was twisted, and they were driving with this kind of signal. It was a single fast spike. The selection of this spike was because it has the characteristic that it contains a whole range of frequencies inside of the signal. So we can later manipulate the signal and find out where we would like to focus the activity. So in this experiment, 4 different dipolar sources, they were placed inside of the phantom, and these sources, these dipolar sources, they were corresponding to brain areas that they present some difficulty to, or covering all the range of difficulties in the detection of the localization accuracy. So we select a source that was superficial, 2 subcortical, 2 sources that correspond to subcortical areas, and also a deep thalamic source to find out whether the MEG can detect these kind of sources. So by filtering the signal in a specific frequency band, what is the interesting and characteristic is that we can create an oscillatory activity, a burst oscillatory activity that resemble the brain oscillatory activity with a specific frequency of interest. So for example, if we filter our signal at the 150 to 250 Hertz, then we have a quite nice burst oscillator activity that can resemble human brain activity with a specific frequency. And by using the MEG, we can record this artificial signal quite nicely. So the signal that we generate with the dipolar patterns and inside of the brain can be recorded quite nicely by using the MEG. So of course you can interrupt any time, and if somebody has questions, like we can go and discuss. Since we would like to find to assess the extremities of the MEG in order to be able to localize the human brain the activity of the human brain, then we select and we focus on very weak signals of the human brain, and these signals are the high frequency oscillations. So high frequency oscillations are signals of high frequency. You can find them in the area of 200 hertz here, and also around 600 hertz. You can see them when you stimulate the median nerve of a healthy individual, and you average data from a high number of trials. And if you filter the data in the specific frequency bands, you can see quite nicely seeing these two frequency bands, the high frequency oscillations. Then what we did with our phantom data, we manipulate and we change the amplitude and the frequency of our dipolar sources here and here and here in order the amplitude to be at the same strength with the physiological signals, in order to generate a signal that seems to be very close to the physiological activity in the human brain. This is the localization results from the phantom data. On the y-axis, you see the localization error in millimeters for the 4 different sources. Here's the localization error for the superficial source, the 2 subcortical sources, and the deep thalamic source. The different colors are different localization techniques. We had beamformer, dipole fitting, the music, and the MFT. And over here on the y-axis, we have the strength of the dipole. So we tested different strengths of the dipole. Where it was, here it's indicated with orange the range of the strength where the high frequency oscillations are located. So the high frequency oscillations in terms of strength, they were around corresponding to a dipolar source of 5 microamperes activation. So what is interesting to see here is that for the superficial source, we saw a quite high localization accuracy of few millimeters, around 2 millimeters. As we are going more deeply in the brain, we see that the localization accuracy becomes worse. We see an activity here of a localization error of 5 millimeters for the subcortical area, also a couple of 5 millimeters for the other subcortical region. And the most interesting is that the deep thalamic source that most of the people believe that we cannot detect with the MEG was detected with an accuracy of around 10 millimeters, 10-15 millimeters. So,
excuse me.
yes?
And your head model here, your experimental model, is saline?
That was a single sphere. We used single spheres. We performed the experimental model with 2 models with a single sphere and multiple spheres. This is the data from the.
What do you mean by multiple spheres?
So each individual sensor it detects activity from a specific sphere. So we have you make the modeling of the head by using different spheres on the brain, and you try to, instead of having one single sphere, you have multiple spheres, and you try to assess to model the surface of the head by doing that.
You experimentally make a different sphere for the skull and a different sphere for the scalp. For multiple spheres, what are your multiple spheres?
So multiple spheres, I had multiple spheres equal to the number of the channels. So these recordings, they were performed with 151 channels. So I had one sphere for each sensor, and the sphere, they were modeling the head shape close to the. So I had 151 spheres, and these spheres, they were trying to fit nicely the head shape. However, here we have a phantom, and in the phantom, the multiple spheres, that they don't make so big difference because it was very close to the ellipsoid.
Okay, always one saline vessel, right?
Yes.
That's all.
Yes.
So this is your fault model is CTF 151.
Yes. Yes.
So it's an axial gradiometers.
Exactly.
So maybe did you try with a magnetometer kind of system?
No. We have tried something similar with a BabyMac here, but with magnetometers never.
And what is the
Not with a phantom.
what
The
is the
gradiometers are planar gradiometers.
No, in the CTF they are axial gradiometers.
Oh, so it's a
Yes.
CTF.
This CTF.
and what are the orientations of the dipole in the simulation? They are very tangential.
Yes, yes, yes. They were tangential. They were close to tangential. I tried to avoid radial sources. Yeah. So the next question is how many trials we need in order to be able to record reliable data. So we perform recordings with different number of trials, 6, 120, and 360. And what was interesting that the localization was quite nice even for a very short number of trials, like only 6 trials. We were able to localize quite accurately the dipolar patterns. The most interesting finding is that if we increase the number of trials and we go up to 360, then the localization accuracy of the make for the deep thalamic sources was quite high, around 10 millimeters. And that was quite impressive. And somebody who's quite skeptical about this, he can say, okay, but this is phantom data. What's happening in the real human brain? This is an experiment that I ran in my previous lab. I averaged a very high number of trials, 3,000 trials, by stimulating the median nerve of the healthy subjects. And I averaged the evoked responses in order to get a very good signal to noise ratio. Then this is what I got from the magnetometers and the gradiometers, and I assumed that the first cortical activity right here at the first peak, the M20 component, as well known in the MAG, should be localized in the contralateral primary somatosensory cortex. But also, if we go around to 14 or 15 milliseconds after the stimulus onset, that we know based on the physiology that the activity is around in the thalamus, then I was assumed that probably the localization of the MAG can reach this activity. And quite surprised that was the case. We performed a similar experiment with a function MRI in order to have a gold standard technique and to compare our findings. This is how it looks like the dipole activity with the MAG, and this is the activity with the function MRI. We have, for the left and the right hand, we see quite nicely overlapping activity of the MAG findings and the function MRI findings. And the most also impressive finding that confirms also our phantom data is that when I have a high to signal noise ratio, the dipole was very close to the thalamus in a distance of around 1.5 to 2 centimeters far from the functional MRI hemodynamic response. So yes, if we increase the number of trials increase the signal noise ratio by average a high number of trials, then MEG is able to localize the activity. So by
no longer phantom, always somatosensory.
This is somatosensory evoked fields.
No
yes,
phantom
no phantom anymore. So by taking advantage of the excellent or very good localization accuracy of the MEG, especially for the superficial sources, we can see quite nicely here in the somatosensory evoked fields elicited by pneumatic stimulation of the fingers. We see how nicely the MEG with different dipoles can represent here the somatotopy, the homoclonus of the primary somatosensory cortex. D1 is the thumb, and D5, so the 5 numbers there, they indicate stimulation of different fingers, and we see how the dipole activity is represent the homoclonus of the primary somatosensory cortex.
Sorry, could you just give us some more details about what you're actually doing? You're doing a single dipole fit? You're allowing
This is a single dipole fit.
Location and orientation to change
Yes.
without any constraints?
Yes, this is a single dipole fit at the peak. Actually, this is not my work, it's a work of my ex-supervisor. So this is a single dipole fit at the first cortical response after pneumatic stimulation of the fingers. So they stimulate the different 5 fingers and with pneumatic stimulators, and then they run a single dipole fitting at the peak of the first activity.
And that was the same technique you used for the thalamic
Yes.
results you showed us before?
Yes, it was single dipole. Yes. Yes, sorry.
How many trials did you have for the thalamic?
3,000.
And you fitted on 14? The peak of 14?
Yes, it was 3,000. So you're talking about.
But you did it for.
You're talking about
14
here.
Oh, 13 milliseconds. Okay. Yeah,
So
no, I know it's
that varies from subject to subject. So in this experiment, we had many subjects, and this in some of the subjects, the activity was localized in 13.8. In some of them, there were 14. I know that depends also on the length of the median nerve.
And did you have, what were your filtering?
That was from 1 to 150 hertz.
Okay.
Can you show the raw data filtered, like a trace filter, and show us a little blip at 14? In other words,
This data has been reported before me by a Japanese group. It was published in Journal of Neuroscience. So this data that
and you can actually see a bleep.
you can see something there.
Yeah.
You can see something there. Probably it's not quite. So you can see something here. It's quite small, but you can see something. In the data of the Japanese groups, it's a little bit more obvious. And they also claim that you can detect the deep thalamic activity, and also you will see later the axial potential propagation. So actually, I tried to replicate these findings from another group. Like I will present later that there are also some criticism about that. Like there's still a lot of work to be done still. So if we're able to detect activity in the primary somatosensory cortex, still the question is that which area is active? Because the assessment of our localization is based on anatomical landmarks, like, for example, the central sulcus. So we still, for example, for this simple experiment, we don't know which area, cytoarchitectonic area, is active, like if it's area 3B or area 3A or 1 or 2. So this information is provided by probabilistic cytoarchitectonic maps. This tool has been developed by Simon Eckhoff, an ex-collaborator. We worked together in Germany. So these cytoarchitectonic maps, they provide a nice tools. For each voxel, they indicate the probability of the voxel to belong to a specific cytoarchitectonic area. So for example, here we have the cytoarchitectonic area of the area 3B, and on red is the high percentance, your activity to be located in the Brodmann area 3B, and the different colors, they indicate the probability. So if you ask how this tool was developed, like I will say in simple words, like they analyze dead brains, like from 10 subjects, they make cytoarchitectonic analysis and they try to find out which is voxel corresponds. And what was quite interesting, by putting the MEG localization data and the probabilistic cytoarchitectonic maps data, is that we try to find out which area is active during the median nerve stimulation. Again, we have median nerve stimulation, always the same experiment. So here is the stimulus onset, somatosensory evoked fields. This is the first cortical response. And then we run dipole fitting at one time point by time point from the peak of the M20 going back. And we see now how the dipole, it's approaching slowly the activity, the heart of the Brodmann area 3B goes up to 70%. And then as we go far from the M20, then we are diverging away. So what is interesting that if we involve in our activity, so the data that are quite consistent, like among many subjects and different runs. Like in some cases we had even very high values, in some cases not so good, like for example 40% or 30%. But the data, they were quite consistent among many subjects. Now if we involve in our analysis neighborhood cytoarchitectonic areas, like for example the Brodmann area 1 and the Brodmann area 2, what we observe that here's again the somatosensory evoked fields. We see the first cortical activity at the 20 millisecond, and then we run again dipole fitting at every single timeline. What we saw that the dipole was approaching the peak of the N20, the Brodmann area 3B, and then as I was going far away, then the dipole was approaching the next area, that is Brodmann area 1 and Brodmann area 2 and Brodmann area 1. So this is quite interesting in the fact that there are animal invasive recordings and also invasive recordings from patients that indicates exactly the same findings. So I will conclude that the MEG so far has a very high localization accuracy under specific circumstances. Under, for example, if you have a very single experiment, if you try to do that with a high cognitive experiment, probably it will not work. So we have an accuracy of 2 to 3 millimeters. When we're talking for subcortical areas, then we can go up to 5 millimeters. And the most interesting is that deep thalamic sources, when we have a very high number of trials, can be localized by some methods and in very simple experimental setups.
Yes, question?
Yes.
So, medium nerve stimulation is kind of the, the strongest legal way one can get brain
Yes.
activity. How do you think this generalizes to other kind of either non-dipolar experiments, or even if you expected a dipolar response, an experiment where you would get a response in the kind of more typical range?
Good point. I was trying to present what it can be done. I don't generalize this in the cognitive neuroscience. If you try to do this in cognitive neuroscience experiment, it will not work. But what I would like to say so far is that MEG can localize deep sources, and MEG has a very high localization accuracy under specific circumstances. And this is simple experiments of neurophysiology. So most of the people, they don't emphasize that. Most of the people, they believe that MEG generally has bad localization accuracy or not so good. And this is what I try to prove, that this is not the case. I will agree with you that we cannot generalize that. If you try to apply this methodology in an emotional processing experiment, it will not work, for sure.
Well, it's also a question of how you define accuracy. I mean,
I fully agree. There are many.
Look at point discrimination. I mean, if you had two dipoles, how accurately could you discriminate them? Is a little bit of a different question. And if you have a single dipole and a very strong signal.
There's a nice work. I don't remember now the name. Like, for example, you have two dipolar sources quite close to each other, or even with a distance of 2 to 3 centimeters. And what you can do, if you run, for example, Monte Carlo simulations, then you can see that the cloud of the activity is much more distributed if your source is much close to each other. So by applying some kind of trying to find out indeed what's happening in a more in a way that you try to retrieve more information, maybe you are able to discriminate that. But it still needs to be a lot of work. Like, and as I said, I try to indicate what can be done, and what's the limitation so far, and what maybe we can go further from here. Yes?
Now, when you say it's an accuracy of 2 to 3 millimeters, what do you say about the co-registration between the MRI picture and the MEG experiment? Are you neglecting that?
It's pity that I don't have another slide. I will tell you my personal experience. I come from a lab in Riken that they collect co-registration data by using 3 different modalities. It was a 3D camera. They capture Minolta camera. They capture the surface of the head. They reconstruct the surface. They collect thousands of points with epochemas data, and they try to bring all this co-registration together. It's a work from Professor Ioannidis. He has published that a long time ago. And when I moved to Europe, the people, they were taking the epochemas data and they would make like tac, 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 and they co-register. So I think that depends on how you run the experiment. So in my personal view, the MEG is a Ferrari, and we should drive it as a Ferrari, and we should not regard it as a cheapy technique that you can do fast things and whatever. So I learned from at least from my Japanese.
You're assuming there's no error of co-registration in the first step.
You assume that you have a minimum error with co-registration.
0.
Let's say one millimeter, like let's say 1 millimeter.
That's pushing.
That's pushing, yes. Yes. Can I? So this is what can be done so far by using the adult MEG systems. And the next question is that by improving the hardware, we can improve also dramatically the localization accuracy of the MEG and the sensitivity. And this is what Yoshi Okada was trying to do by building the baby MEG system, the baby squid, as he called it. So the idea was quite simple. If we bring the pickup coils much more closer to the headrest, and if we have a higher density of coils in the sensor array, then we may be able to improve dramatically our localization accuracy and the sensitivity, especially in cases that we record data from children where their scalp and the skin is quite thin compared to the adults. So this is how it looks like the anatomy of our system at Waltham. We have here the sensors, 76 sensors here in the headrest. So the headrest is here. Then we have here the squid electronics. the squid electronics, they control the squids, and they transfer the data from the squids up to the analog to digital conversion cards. Then through a fiber optic, the data are transferred, displayed, and stored outside of the shielded room. So it's a quite simple construction. And we are trying now to find out how this works and if we can improve our localization accuracy and our sensitivity. So for people who have never been to Waltham, probably most of you, like the system is accommodated in a single layer a single layer shielded room. Then the babies or the children, they lay down on the bed, they place their heads here on the headrest. The headrest consists of 76 gradiometers, and the co-registration, I will give you more details later, is performed by a stereoscopic camera here. So this is how it looks like, the sensor array of the BrainMap inside. So we have pickup coils with a diameter of 5 to 6 millimeters. We have the distance between the coils, the mean distance between the coils is 14 millimeters. And we have 76 in our system. In the next system, we have in a year from now, we'll have a high number of sensors. I try to give an idea how much more dense is compared to, for example, the commercial systems. Like here, this is how it looks like, the coil of the Electa. We have 28 millimeters. I will not call it diameter, but I will call it the distance between the edges of the two coils. And we have also a distance of 34 millimeters between the center to center between the two coils. So the BrainMap has a much more dense sensor array compared to the adult commercial systems. And we are now in the process, we cannot claim still, but we are in the process to find out if this indeed improves the localization accuracy. So this work was done by my technician, Shiran Dosi. He did most of the work with help of Mati. We stole his his fandom, the dry fandom. So we secured the fandom here on the BrainMap. Also, Mati helped us a lot with trying to find out what exactly should be done. This is the localization result, single dipole. Again, this is the sensor array. On red we have on blue we have on red we have the actual location of the dipoles. So the location of the dipoles and the orientation. And on blue we have the estimated location of the dipoles by using the dry fandom. What is quite impressive is not that I was expecting. We have a localization error of 1 to 2 millimeters. Again, ignoring the co-registration error, though it's a manual procedure, like we did real co-registration, simulation data. And what's more impressive is that we have sources quite deep, far 8 to 9 centimeters far from the sensor array, that were localized with an accuracy of 5 to 6 millimeters. So again, I don't claim that you can do that with humans, but this is what my fandom data, actually Shiran's fandom data, they indicate. So
a dry phantom, like there is no volume conduction.
Yes, I agree. You cannot make pure projection of your findings. Just I present what can be done. Yeah, go on.
So this
Kill it.
So this phantom exactly produces the same magnetic field as a dipole in a spherically symmetric volume current. So
Okay.
the effect of volume current,
I didn't know that.
the volume current is exactly
Okay.
simulated by the phantom.
So, of course, so far I was discussing about hardware and software and all this kind of stuff. But the BrainMap facility at Waltham, it's more than that. We have a facility there designed and established exclusively for children. And that's our role of the whole group there. We are trying so far to be friendly with the patients. We have to make a very children-friendly environment with different colors on the paintings and different toys. And always we're trying to be smiley to the people. Like this is how it's the BrainMap facility. So it's more than a prototype system running there. So this answers the question of the co-registration. We struggle a lot to make this co-registration procedure. So what we usually do, we collect data by using the Pohima scale digitization system. We collect very high number of trials, very high number of points, up to 1,000 sometimes, in order to reconstruct accurately the scalp of the children. In addition to that, we mark 15 points with a watermark pen, and then we digitize these points too. And we digitize the same points inside of the shielded room by using the Polaris 3D camera. So by doing that, we are trying to reconstruct the head shape by using the MRI, also to fit all this information by using the Pohimas data, and also to bring all this information to the same coordinate system, the sensor, the functional data, and the anatomical data. The discrepancy you see here is because we usually, and sometimes, we don't have the same MRI of the children. So if you have, because most of the children, they have an MRI, for example, of months old or even a year. So we try to do our best by bringing a template of some other child, or in this case, for example, this MRI, it was a couple of months previous than the recording. So this answers the question about the co-registration error, where you are at the co-registration error indeed. So this is how we perform also recordings of simultaneously also EEG data. We place the cap of the 128 channels usually. We digitize the points of the position of the electrodes. And this is what's happening inside of the shielded room. We place the child in the position that the sensor array, the partial coverage, should focus on a specific area. Then we digitize the same points that we digitized before. This stylus pencil. At the beginning, when we're describing all this procedure and all the group was discussing whether this will work and the kids that they will move all the time, we were pretty sure that this will ever work. But indeed it works. Like, we try to make a fairy tale that this magic stylus, it measures your activity or whatever, and the children, they don't move. Like, it's quite impressive. So also, this is not one man's job. There's a whole group there of many people trying to distract the kids what they're doing during the position of the electrodes and all this kind of procedure. And it's a kind of tedious job, especially with children that they have behavioral problems. And we had some cases. So today I will present the data from a patient with epileptic seizures and tuberous sclerosis complex. It's a genetic disorder that produce many tumors inside of the brain and also in other parts of the human body. And I'm not specialist in epilepsy, but I start to like it a lot. What we're trying to find out is to try to find interictal spikes and to localize them for this specific patient. So we record 128 EEG channels at the same time with the BabyMap recordings. And the first thing we did, we tried to identify the spikes and in both EEG and the MEC, with help, of course, of somebody who's specialist. So I will not go in details with the analysis because we don't have run out of time. So this specific patient, they had a big tumor in the right parietal cortex, and the epileptologists, they were assuming that a lot of activity will come from there. So what we did, we identified on the EEG signal, here's the EEG channels, the different channels, the spikes. The spikes, they were much prominent in the P8 and the P4 electrodes, the EEG electrodes. Then we select spikes that they have always the same spatiotemporal pattern on the scalp in order to be somehow sure that we don't average irrelevant things. And then we average the signal to increase our signal to noise ratio. This is how it looks like the signal, the average signal.
The shape, going back to the other slide, let's look at spike 2.
Sorry?
Next slide. There.
Yes.
Look at spike 2 in the upper left. There's a
Here?
funny shape. What is the shape? The whole shape.
This is,
Yeah, explain that.
how is the reconstruction?
No, but it's sort of like a thing. Is that supposed to be the child's head or what?
No, actually we project, this is the, good question. This is the EEG electrodes placed on the co-registration of the MEC. And why it's distorted? Because we have not made so many cups, and the cups sometimes they don't fit perfectly to the subject. So like the EEG electrodes, they are not perfectly fit on the subjects.
Roughly the shape of the cap.
Roughly, yes. So for example, here you are in the same space, but you see that our spatial This is our sensor array that offers partial coverage, and this is our main limitation. We cannot have full recordings of the head at the same time. So, so this indicates the special. So this indicates the sensor array. So Matihis has done some kind of good work on with the M&E software, but I don't have this data here to present. Like because in the M&E software it looks different. Like anyway, so so at the same proximity of the EEG spikes, we went at the same time point, and we tried to find whether there are make spikes there. That was not always the case. So we select spikes that they were also both in the EEG and both in the MEG. And we select the peak of the spikes, and then we average the data. Always have the same spatio-temporal pattern on the skull. And this is how it looks like the signal-to-noise ratio for these patterns. Then we localize the activity, both average signal and single events. Let's say this is a minimum error estimation by using the brainstorm. We have 22 spikes. This is the average of the 22 MEG spikes, and this is how it looks, a single spike. I select a good one. This is the localization of the minimum error estimation. On white, you see the tubor location, and you see that we have a quite consistent activity, a phylogenetic activity very close to the tubor. The dipole fitting also gave us very similar results. Here's the dipole fitting on the average signal. And this is the source localization of epileptic spikes. I compare here the results of the MEG signal with the localization of the EEG. Here, how it looks like the average MEG spikes, and here's how it looks like the average EEG spikes. What was interesting, probably people who know about epilepsy, they will not be surprised by that. But when the peak of the MEG activity corresponds to actually the maximum change on the signal to the EEG, and at least to my knowledge, like this seems to be consistent with physics. Like a maximum change in the electrical field generates the maximum magnetic field. This is the localization results from the baby MEG. This is the localization results from the EEG, much more blurry, but always indicate the same location. What was quite impressive is this analysis of two different recordings. The same patient came two different times in our lab, and the data are analyzed by two different persons individually, me and Alex Hunold, who is now in Germany. And this is the results. Recording of day 1, recording of day 2. This is the extraction of the tubor. This is extraction of the tubor. Different spikes, like the two different persons, they identify the spikes, and we see how replicatable are the results of the localization. Like 9 millimeters far from the surface of the tubor, and 2 millimeters far from the surface of the tubor. Like on blue here, you see the dipole fitting at the peak of the average activity, and then on red you see some of the individual spikes. So, any questions so far? So I will try to go to the next work that I was doing in my previous lab to give an overview of an experiment in the functional connectivity. This work has not been published yet. So since so far we were trying to focus on the functional segmentation of the human brain to see which, in both pathology and both healthy brain, like to focus on which area is doing what. The next step is to see how these areas, they are talking to each other, how they communicate. And this is what we're trying to do with the functional connectivity in the MEG. So this is a study that was performed in Tübingen in Germany. What the subject they had to do, it was a visual motor integration task. This task possessed a demand on the brain to correlate the activity, the visual cortex to correlate the activity with the motor cortex, and to find how they are communicate to each other. So the subject that was sitting in the MEG, this is an adult system. What we had here, we had a rectangular window changing in size all the time, continuously, and then the subjects, by pressing and by modulating their strength in this pin script, they were trying to keep all the time, as much possible they can, the window very close to the reference window, the black one. So this is data from one subject, how good it was performing. On black is what it should do, and on red is what the subject was trying to do. And in order to test the specificity of our experiment, what we're trying to do is that we're trying to check the specificity by testing also two control conditions, pure motor condition, the subject without getting any visual input. They were trying to just move or press the pinch grip, and the pure visual condition where the subject were not doing nothing here. They were just trying to watch simply the target that was moving randomly. So this is, we use the beamformer, like I will not go so much into details. We ended up to pseudo T-maps that indicate increase and decrease of the power of the signal compared to the rest condition. So we compared the active condition when the subject they were performing the task with the rest. Then we brought all this, all the functional data normalized to the MNI space, and then we ran statistics across all subjects. And for the regions of interest, then we estimate the virtual channel by using the beamformer. So this is how it looks like the localization results. The different colors you see here is the different probabilistic cytoarchitectonic maps. I try to use this tool because it gives you an idea where your activity is localized. So this is the areas that they were active during the visual motor integration task. We saw many areas to show up. As it was expected, earlier visual areas in the primary visual cortex, we saw activity in the parietal cortex, the Brodmann area 3B, somatosensory, motor areas, the contralateral S2, and also quite interesting, what we found interesting, it was the inferior frontal gyrus that was show up in bilaterally only during the experiment. Like, because I was naive with this kind of region, like what this region is possible for, I knew that IFG and these areas are responsible for language. Then I found out that there's some literature reporting that IFG also is responsible for the control of fine motor movements, and this is also what we are doing when we are speaking, that our brain controls fine motor areas when we are speaking. So when I discussed this with my ex-supervisor, he found quite interesting that this could be the case, that it's an area that controls fine movements of the muscles. So then we're trying to identify a network that started from the primary visual cortex, that is the input of the signal, to the output, that was the primary motor cortex, and we try to find a network from the input to the output. And for these regions of interest, what we observe a decrease of the power for the alpha and the beta band. This is the power signal for the left V2 region of interest. This is for the left IFG, and this is for the left motor cortex. We saw the beta band also, there was some change there, but it was not specific for the visual motor integration task. The reviewers, they asked also that maybe this is of interest because the alpha modulates also with the difficulty of the task. It could not be necessarily specific to the task of visual motor integration. But surprisingly, like the alpha band, they never, the beta band, they never saw some kind of specificity for this task. What was interesting, we see a suppression of the alpha in a gradual way, I would say. So here is the rest condition on blue. On red is the visual motor integration task. In the middle, we see the control conditions. And this was for most of the areas or areas of interest. But what was nicely is that we see an increase of the coherence in the alpha band. It was mostly alpha band and also a little bit lower frequencies. But we focused on the alpha band because the power gave significant results for the alpha power. So while the power was decreasing, then in the alpha band, then the coherence was increasing in the same frequency band, and that was indicate a functional connectivity, a coherence connectivity between the motor, the visual area, and the IFG, and then from the IFG to the motor cortex. So by focusing on areas, trying to find the link, how the information flows from the visual cortex to the motor cortex, we saw on red is the functional connectivity, significant coherence data. These dust lines is that when we have the p-value was very close to 0.05. So marginally significant findings. We see that a connectivity between the visual cortex, the IFG, and the motor cortex in the contralateral side always, for both the left IFG and the right IFG, and that was specific only for the visual motor integration task. It was not for the motor and not for the visual task. So the conclusion of this study is that IFG seems to play a role of integration area for the visual motor in a visual motor integration task. We're trying to publish this paper now in cerebral cortex. And we have some criticism from the reviewers about the specific frequency bands, like whether other frequency bands like the gamma band is also involved in the story. Yes?
Was that alpha?
That was alpha. Yes, that was alpha.
You didn't find anything for beta?
Not in the coherence. So the next step is whether we are able to see information flow from one area to the other one. We know that these areas are talking to each other, and the question is that whether we know whether we can with MEG to track the information flow from area A to area B. This is a study that was published this year. I was based on a paper published before in Journal of Neuroscience from a Japanese group, as I said before. I forgot now the names. They report exactly the same findings. What I added DTI. So it's quite impressive. Like, and I'm still not 100% sure of whether the findings it's really true. That's why I try to make many tests to check what we can get from this data. So, as I said before, like we managed to localize the activity in the deep thalamic sources at 14 milliseconds after the stimulus onset. Always median nerve stimulation. So here's average data 3,000 trials. And this is how the dipoles they look like when we run dipole fitting from the every time single point, every sample. And this is how the dipoles they go, they follow this path. So you see that the path pass from the area of thalamus, that is around here, up to the Brodmann area 1, like the primary somatosensory cortex. And what was nice and impressive, like it follows very close, I will not say perfectly, but very close, the thalamocortical fibers, from the thalamus to the S1. I was very, when I published this paper, I was very limited in the DTI. I was just doing the DTI by myself, like very naively. Now I know that people, they are doing DTI quite nicely. I was not able to track some fibers because there's some intercrossing there. So there are improvements, especially in the DTI part. So in order to check the reliability of our findings, I ran Monte Carlo simulations to see the region, how reliable is our recordings. So we see that a horn was created, indicate that there's more error or localization error for the dipoles that they were deep inside. And then as we were approaching the primary somatosensory cortex, then we are going to a more closed cloud. Always very close to the thalamocortical fibers. Then my ex-supervisor was very skeptical. He told me, okay, the guys in the Japanese group, they published that, but I'm not sure whether we can record action potential propagation across the fibers. So he told me, let's check that. Like there are two sources. One source is in the primary somatosensory cortex, the other is on the thalamus, and these sources they are activated with an overlapping pattern. So maybe this is what generates this activity. So this is what we did here. We placed two dipolar sources. One supposed to be in the S1, one is supposed to be in the S in the second area. Like sorry, this is the S1, this is the thalamus, and then the two sources they were having an overlapping, shifting overlapping activity, and then we ran dipole fitting on this generated simulation data. And then the dipoles they were moving on a straight line, while what we see here we had the curvy path with the real data. Somebody can say, and this is what my supervisor was telling me, there's another source here, and this source attracts this. So, of course, you can play thousands of times with this data. And then we end up to the solution that we have the ideal test for our case. And the ideal test is this patient. So this patient has a perinatally acquired periventricular lesion in one side. So we see the lesion here. I met this person, she's German, she studies philosophy in the University of Tübingen. She's perfectly healthy, or she looks healthy, and when I saw these results I was shocked. So this also indicates the ability of the brain to have plasticity. And so the test was the following. We ran the same experiment, and we expect that, okay, if it's a modeling error, then what will happen? The dipoles, if it's a modeling error, will drive from here and will go inside of the lesion, and also the same for the healthy part. And this will kill our story. But that was not the case. So this is how it looks like, the fibers. We didn't manage to track the fibers from the pathological, the lesion site, like this way. This is what we're trying to manage to do with the DTI. Also, a part that was missing here. Like as I said before, I'm not a specialist on the DTI, so there are people who can do much better job with that. And this is how the data, they look like, the DTI data for the, we see here that the fibers, they follow, they are very close, but they deviate the lesion. And then this is how it looks like the dipole data. So this is how it looks like the dipoles. They're moving from the healthy part, not very close, I would say, to the thalamocortical fibers, but following the same path quite nicely and quite consistently with the healthy subject. And this is how it looks like the data from the other side. This is primary somatosensory area here. So there was no lesion. There was some jump in the dipoles. We don't know why. Probably the sources, they were radial and we don't detect them. But this is what we found with the patient. You can see it much more nicely here. You can see it much more nicely here. This is healthy side, how the dipoles are running on the healthy side. This is how the dipoles they run on the pathological side. There's no deviation, there's no invasion inside of the lesion. So, of course, there are many questions about this work, in the sense that, that's why also when I wrote the paper, I state on the title a question rather than a statement. Like, for example, what I don't like is the dipoles orientation here. If it was actual potential propagation, then the dipoles, they should indicate like an orientation from downstairs, from down to the top. Like, but that was not the case. And there was a discussion also which kind of neurophysiological mechanisms can generate this data. Like whether there's activity that is cancelled to each other and what remains. Okay, so I will close with I have worked with MEG so far, and I believe that MEG is the ideal tool, especially for studying functional connectivity. It's also a very safe tool and convenient tool, especially for children's studies. And it's a safe and efficient way to study the human brain development, especially in children. So this is our team here in a children's hospital. Like Matti, I don't know if you like this photograph out from the internet. Thank you very much.